Okay, welcome back. So now we are going to discuss about continuous culture systems. So let me summarize. So we discussed about the bioreactor. How does a bioreactor look like, and what are the accessories of a bioreactor, and what is the classification of bioreactors based on their size. After that, we had a look at what are the bioreactors classification based on their mode of operation. So till now we have discussed about two important modes of operation of bioreactors. First one is called a batch mode of operation, and the second one is called a fed batch mode of operation. So even till date, batch mode and fed batch mode are two most widely used modes of operation for producing biological products. And the third mode of operation is called continuous culturing mode or continuous culturing systems. So if you have a look at the schematic representation, the top part looks exactly same as batch mode. If you consider this thing, looks like a fed batch mode where a feed tank is connected, and the only additional change that we can see with a continuous culturing system comparing with the fed batch mode is we have a product tank here. Okay, so in case of your continuous culturing system, the volume of the broth or the volume of the broth inside the bioreactors should be kept constant, right? The working volumes of the liquid or the broth that is there inside the bioreactor has to be kept constant. So that's the important condition that we need to maintain for making the cells grow in a continuous culture system, right? So volume, as I said, volume has to be kept constant inside the continuous culture system. What does that mean is we are going to send the feed continuously into the bioreactor. So what happens if we send the feed continuously into the bioreactor? The volume gradually increases inside the bioreactor, right? So it can have undesirable effects. So we are taking continuously feed out from the bioreactor. So ideal condition is the flow rate that is going into the bioreactor should be exactly the same as the feed that is coming out or the product that is coming out from the bioreactor. So the input flow rate should be exactly equivalent to the output flow rate. Then only the culture volumes can be maintained constant inside the bioreactor. So with the help of a feed tank, we are slowly sending the feed in, and with the help of an output tank or a product tank, we are collecting the product out. So here you might get a doubt. How can we get only the product out? So this is not the only product that is being collected. The biomass as well as metabolized products, unwanted substrates along with the product will be coming into this product tank, and here we are going to collect the product and do the further downstream processing of this liquid that we have collected and separate the product from the broth which is collected. So let's have a look at what are the advantages of the continuous culture system. So the first major advantage is productivity and growth rate can be optimized, right? So we can enhance the productivity, we can enhance the growth rate by using continuous culture system. So why? Because we can continuously culture these for several months of time. So we can culture the microorganisms for several months by keeping the organisms in physiological steady state. Okay, physiological steady state is an important parameter which need to be discussed whenever you are talking about continuous culture systems. I'll explain about physiological steady state when we are discussing about chemostat and turbidestat in detail, right? So right now you remember that. So organisms which are growing in a continuous culture system are in physiological steady state. That means no parameters can affect the growth rate of the organisms which are growing inside the continuous culture system. Only parameters that can affect the growth rate of the microorganisms growing inside the continuous culture system are nothing but the environmental factors. The temperature, the pH, and other changes, environmental factors which are acting on the bioreactor only can change the growth rate. So this one major advantage is of running the culture in continuous culture system is we can clearly understand the effect of the environmental factor or the physical factors can be easily analyzed and studied. Why? Because no other factors will have an effect because all the other parameters are kept ideally constant. Starting with the substrate, amount of carbon source, amount of nitrogen source, and the amount of the culture that is growing, it will be maintained exactly the same. So the only parameters that can affect the growth rate is the environmental factors acting on the bioreactor. Second major advantage, longer period of productivity with less downtime. So we have seen in the batch system, after every batch we need to do the cleaning, sterilizing, autoclaving, all this stuff, and before we go for further production with the batch. Whereas in this case, we can continuously use the same reactor for several months of time and keep producing the product. That's one major advantage, and it can take advantage of cell immobilization. So we can immobilize microorganisms inside the bioreactor, continuously send the feed in, make the immobilized cells to utilize the substrate and convert the substrate into product. That product will be going into the product tank, and again media will be going in. Organisms will stay there in an immobilized form and they will be helping in producing the product. So this can also be achieve high production with a continuous culture system. So these are some of the advantages, and another advantage is evolution in these cultures can be studied. Since we are growing the organisms for several generations in the same vessels for several months of time, we can easily understand or perform the evolutionary studies with the microorganisms. So what are the disadvantages? Are there any disadvantages of continuous culture system? First major drawback is any product that is produced by continuous culture system will not be approved by US FDA. Okay, US FDA is a governing authority which will regulate the production of therapeutic or bio products. If any bioproduct or therapeutic product or drug is produced using a continuous culture system, these such kind of drugs will not be accepted by US FDA. The major reason behind this is we cannot tag a batch number for the products that is coming out from the bioreactor. That means if something goes wrong with the product that is coming out from a continuous culture system, we cannot trace back where it went wrong, right? At what point of time the product has came out. So we don't know, we can't find out. We can trace back the time or the batch at which so we need to discard the entire production process, right? That's a one major drawback. And since we are not able to locate or trace back the product where it went wrong during the production process, so such kind are not considered as current good manufacturing practices by US FDA. So continuous culturing system is any drugs produced by continuous culture system is not approved by the US FDA. Next one, not all products are produced optimally in continuous culture system. So let's say there are some products which require stationary phase or enzymes from stationary phase, or some products which require enzymes from growth phase, right? So let's assume a product X, which require both the enzymes, an enzyme from the stationary phase as well as an enzyme from an exponential phase. So if such kind of products are there which require enzymes from different phases, such kind of products cannot be optimally produced, because in case of continuous culture system, all the organisms which are growing are ideally present in the same stage. So if they are growing in the log phase, all are in log phase. More ideally, all the organisms are there in the exponential phase as long as the culturing is continued. So if you want enzymes from the different stages to help in producing a product, it may not be feasible. So such kind of products are not optimally produced by the continuous culture system. And non-growth associated products such as antibiotics and monoclonal antibodies are not produced well in continuous system. So we have seen for any non-growth related product production, the ideal mode of operation is batch and fed batch. Why? Because this will lead all the organisms to move into the stationary phase at one point or another point to produce the product. Since stationary phase is ideally not achieved in the continuous culture system, continuous culture system is mostly used for growth related products. That means products which are produced during the growth phase, like your primary metabolites. All the primary metabolites are produced using continuous culture system, but secondary metabolites are not optimally produced by continuous culturing system. Right? Another major drawback, since this is not a closed system, it's an open system. We are sending continuously feeding and continuously taking out the product out. There are chances of contaminations inside the vessel, huge disadvantage, and always the mutation problem. Since we are growing the organisms for a longer period of time, culture may get mutated, and the genes which you have incorporated into the plasmid, gene of our interest, recombinant gene of our interest, which produces the product of our interest, may be shredded off from the culture, or the back mutation may happen during the course of time, which leads to non-productivity, right? So these are all things can happen. So these are some of the downsides or disadvantages of continuous culture system. I'll wind up this session here. Thank you.